Hi everyone, I'm Xiao from CSBL. My presentation title for the Journal Club is Patterns in Soil Microbiodiversity Across Europe, which published on Nature Communication in June this year. And I chose this journal as reference to lend the ideas to analyze and write the metagenomic related papers. Here are the highlights of the journal. In this journal, researchers investigated factors that not only driving microbial community composition and diversity, but also have the relationship with microbial functioning. They analyzed potential functional groups along a gradient of increasing land use perturbation, detecting 79,000 bacterial and 25,000 fungal RTUs in 715 sites across 24 European countries. The lowest bacterial and fungal diversity in less disturbed environments compared to grasslands and highly disturbed environments. And highly disturbed environments contain significantly more bacterial chemoheterotrophs, fungal plant pathogens, and saprotrophs, and have less beneficial fungal plant symbionts compared to woodlands and grasslands. Spatial patterns of microbial communities and predicted functions are best explained when interactions among the major determinants are considered, such as vegetation cover, climate, and soil properties. And at the end of the journal, they also proposed guidelines for environmental policy actions and argue that taxonomical and functional diversity should be considered simultaneously for monitoring purposes. In this study, researchers analyzed the soil microbial diversity considered in community level and the functional level. At the community level, land use perturbation has been identified as one of the main anthropic pressure affecting soil microbial diversity, resulting in community composition shifts. Other reported factors influencing soil microbial communities are climate, soil properties, and vegetation. Changes in climate and in specific soil properties, such as pH, texture, and nitrogen availability, also lead to changes in microorganism assemblages. At the functional level, land use perturbation may alter the composition of soil functional groups. And before the study, researchers hypothesized that land use perturbation affects soil microbial diversity and community structure and potential functional groups as observed for the above-ground biodiversity. Second, bacterial and fungal guides and potential functional groups are shaped by different forces. Last but not least, interactions between vegetation cover, climate, and soil properties are more important and informative than single effects in driving the assembly of bacterial and fungal soil communities and potential functional groups. In this study, researchers analyzed DNA sequences of two groups of microorganisms from 715 soil samples collected from 23 countries of European Union and United Kingdom. And soil sampling is performed within the framework of the soil module of the LUCAS. Sampling locations cover both semi-natural and highly managed environments, including 6 vegetation cover types subjected to an increasing land use perturbation gradient. And the breaking through point of this study is that none of the published studies so far has explored compared bacterial and fungal functional groups among the full range of vegetation cover types and associated land uses, preventing an assessment of the impacts of an increasing land use perturbation on targeted functions associated to soil microorganisms by affecting plant diversity and altering carbon and nitrogen retention. In this study, researchers analyzed the datasets to observe the microbial diversity, community structure, and functional groups of bacteria and fungi. For microbial diversity, the observed RT richness and the Shana index were calculated as a measure of bacteria and fungal alpha diversity. Alpha diversity is a measure of microbiome diversity applicable to a single sample, while beta diversity is a measure of the similarity or dissimilarity of two communities. And the dissimilarity matrix was calculated to assess the diversity in OTUs between sites using a Hallinger transformation, and the beta diversity also analyzed by distance-based redundancy analysis. The relationship between microbial functional groups and the investigated drivers was visualized by means of ordination pie plots. Here are the results of the study. First part is land use perturbation effects on microbial richness, diversity, and functional groups. The entire dataset included 79,593 bacterial ZOTUs and 25,962 fungal OTUs. The observed OTU richness and Shannon index were calculated as a measure of bacterial and fungal alpha diversity, and the results show that the observed OTU richness and Shannon diversity index were lower in woodlands and compared to grasslands and croplands for both bacteria and fungi. Also, vegetation cover had a significant impact on bacterial and fungal community structure, as shown in figure E and F. The highest difference in community structure was found between microbial communities in croplands and woodlands. The distribution of interfered bacterial and fungal functional groups also differed among vegetation cover types. Bacterial chemoheterotrophs dominated in croplands and intensive grasslands. Nitrogen-fixing bacteria were more abundant in woodlands as well as in extensive grasslands. Bacterial pathogens dominated in coniferous forests. And regarding fungi, ectomycorrhizal symbionts dominated in woodlands, especially in coniferous forests. Vascular mycorrhizal fungi were more abundant in grasslands, particularly in extensively managed ones. Fungal saprotrophs and plant pathogens were dominant in croplands and grasslands. A total of 65,206 bacterial ZOTUs and 7,642 fungal OTUs were not assigned to any functional group, representing 81.9% of all bacterial ZOTUs and 29.4% of all fungal OTUs. The second part of the result is the single effects of soil properties, climate, and vegetation cover. For the single effects on microbial alpha diversity, variation partitioning analysis showed that the bacterial communities were mainly driven by soil properties. 14.5% unique variation explained for the observed richness, followed by climate and vegetation cover. Conversely, fungal communities were shaped by vegetation cover most, then soil properties and climate. Considering beta diversity, these three factors have similar effects on bacterial community composition on their alpha diversity, while for fungi, soil properties had the strongest impact on fungal community composition, followed by vegetation cover and climate. Researchers also performed variation partitioning analysis to assess which variables best explain microbial functional groups, focusing on single factors. Here, bacterial or fungal variable importance for each soil properties and climatic variable in explaining the proportion of OTUs belonging to a given functional group. For next step, they used an ordination method and associated bioplots to illustrate how community diversity and structure and presence of potential functional groups were preferentially related to certain combination of vegetation cover type, soil properties, and climatic variables. Variation partitioning showed that the most relevant interactions shaping bacterial and fungal richness diversity and community compositions were the ones involving vegetation cover. In particular, interactions among soil properties and vegetation cover represented 4.8%, 4%, and 2.8% of unique variance explained for bacterial and fungal observed richness and fungal diversity respectively, as shown in figure A and B. Those interactions mainly explain the differences in community composition between sites for both bacteria and fungi. For more detail, figure C to I show the variable importance of the first 5 two-way interaction terms in the interaction models for bacterial and fungal functional groups for bacterial camel heterotrophs, bacterial nitrogen fixer, bacterial pathogens, ectomycorrhizal fungi, avascular mycorrhizal fungi, fungal saprotrophs, and fungal plant pathogens. Overall, interaction models performed better than the single effect ones for both groups of microorganisms and inferred functional groups. Interactions models explained from 2% up to 16% of additional variance for all considered metrics and displayed smaller archaic information criterion values. This journal considered and discussed the results with following aspects. They are aspects of land use perturbation of microbial communities, effects of land use perturbation on functional potential, effects of land use perturbation on biodiversity ecosystem functioning relationship, single drivers of microbial communities, single drivers of functional potential, interactive drivers of microbial communities and the functional potential, study limitations and perspectives, and at the end, they proposed guidelines for policy-related applications. In conclusion, this study demonstrated that the microbial diversity, community structure, and the potential functional group distribution vary along a gradient of increasing land use perturbation. At European scale, microbial communities from more disturbed areas differ the most from communities associated to semi-natural habitats. Soils harboring richer and more diverse communities are also characterized by a higher presence of potential fungal pathogens. In contrast, woodlands and extensive grasslands harbor more fungal plant symbionts and nitrogen-fixing bacteria. Investigated 3 types of environmental drivers: soil properties, climate, and vegetation cover. And the results show that bacterial diversity is mainly shaped by soil conditions, while fungal diversity is mainly influenced by vegetation cover. And differences in microbial community structure among sites are better explained by variation in soil properties. Distributions of inferred microbial functional groups are shaped by different types of drivers, and that the same soil and climatic conditions can have opposite effect on groups. Thank you for your listening.